So one of the really wonderful things about being part of the program committee for this meeting is we got to sit around and think about if we could plan our dream day of who we wanted to be inspired by and learn from. And so many people that we invited said yes. It was really amazing. And our next panel continues that theme. We're going to move to innovation. So we've got a panel of 4 wonderful engineers. I'm going to very briefly introduce them. Their bios are in the program. Then they'll each present, and we'll have an opportunity for Q&A at the end. So from my left to right, we have Kelsey Swingle, who is an assistant professor in the Department of Bioengineering at Rice University. We have Giuliani Sempianato, who is an assistant professor in electrical and computer engineering here at Rice. Kevin McHugh, who is an associate professor in bioengineering at Rice, and Rahima Ben-Habor, who is an associate professor in the UNC-NC State Joint Department of Biomedical Engineering. And they're all going to talk to us about innovations in maternal and child health. And I think we are starting with Kelsey, if I am not wrong. And I'm going to go ahead and turn it over to you.
Thank you. Thanks, Rebecca, for the opportunity to speak. Really excited to be here with you all today. So like Rebecca mentioned, we're a team of engineers. So I'm going to tell you a little bit about my lab's work towards trying to develop drug delivery technologies for treating pregnancy disorders like preeclampsia. So I, of course, don't really have to set the stage for this audience. You all know, and we've heard a lot today about the reality of obstetric health, of maternal health here in the U.S. I, of course, with today's conference focus of global to local, want to emphasize what we've heard a lot about at this idea of maternal care deserts, right? Specifically here in Texas. So my team, my lab here at Rice, we're scientists, we're engineers, we're thinking about how we can tackle some of these underserved pregnancy disorders like preeclampsia. So I just very high level want to provide an overview of preeclampsia, a leading cause of maternal and fetal morbidity and mortality. Primarily diagnosed as an hypertensive disorder, so preeclampsia is diagnosed by the sudden onset of hypertension during pregnancy, but it's a really complex multi-organ disease. So the schematic is pretty intentionally overwhelming. It's trying to illustrate all the organ systems that preeclampsia can affect. Patients can be at risk of developing seizures, risk of elevated liver enzymes, kidney injury. Fetuses from these pregnancies are often growth restricted because they're not receiving enough nutrients and oxygen. So standard of care for preeclampsia is focused on symptom management, so antihypertensives to manage blood pressure, anticonvulsants to manage seizures. Sorry about that. But the challenge here is that there are no therapeutics available in the clinic that will slow or stop disease progression, right? So as we think about trying to actually tackle what's going wrong in preeclampsia, we have to think about the underlying pathology. And preeclampsia is predominantly a placental insufficiency disorder, right? So we look at this tissue, this placental tissue, it's clearly a highly vascularized tissue. Its main function is to facilitate the exchange of nutrients, oxygen, and waste between maternal and fetal circulation. Now that's great if the placenta is acting how it's supposed to, but in preeclampsia, things are clearly going wrong. We can see from this inset schematic that preeclampsia is characterized by improper vascularization in the placenta. We instead have a low blood flow environment. When we look at the molecular level, instead of having a highly dilated tissue with proper blood flow, preeclampsia is characterized by upregulated levels of sFlt1. This is the soluble form of the VEGF receptor. So it's sequestering VEGF, vascular endothelial growth factor, a really important growth factor for vasodilation of the placenta, right? So sFlt1 sequestering VEGF in circulation, preventing it from acting on the placental vasculature, and we just kind of get this positive feedback effect. So as a team of engineers, how can we think about actually creating a therapy that can induce vasodilation in the placenta to treat preeclampsia? So my lab works on ionizable lipid nanoparticles as a clinically approved drug delivery technology for nucleic acids. This delivery technology has undergone quite a bit of evolution over the last several decades by some very notable scientists. LMPs were first FDA approved only a few years ago in 2018 for treating a rare liver disease. And of course, many of us are probably familiar with ionizable lipid nanoparticles for their utilization in the COVID-19 vaccines a few years ago. Now, if you take any of these off-the-shelf technologies, these FDA-approved LMPs, they're going to tend to accumulate in the liver due to a first-pass hepatic clearance effect. And of course, in our case, that's not that helpful. We want to treat preeclampsia. We need to engineer a technology that's actually going to enable nucleic acid delivery, like messenger mRNA or messenger RNA, to the placenta, right? So how can we actually engineer these technologies to do what we want them to do? One of the great things about LMPs is that they're highly tunable. They're very modular. We can very easily tune them to achieve tissue-specific nucleic acid delivery. So here's a schematic of how we make LMPs in the lab. We combine our lipids with a nucleic acid like mRNA, mix them together and form LMPs. The way in which LMPs are modular is that we can alter the structure of any one of these lipids, or we can alter the relative amount of these lipids to one another, which means that LMP composition you can change it in essentially infinitely many ways, right? So this is a big focus of the LMP field over the last several years, is this idea of extrahepatic delivery. How do we tune LMP composition to achieve delivery outside the liver, like to the lung or the spleen? And about the time that we started this work, the placenta was a completely novel tissue target for LMP-mediated nucleic acid delivery. So in my lab, we've opted to use some high-throughput screening technologies that allow us to test really large design spaces of LMP formulations, so we can identify one or multiple that achieve delivery to the placenta. So I'm going to do a very high-level overview of some of the kind of LMP work here, and instead focus more on some of the preclinical data we have in some mouse models of preeclampsia. But this high-throughput screening technology allowed us to evaluate a large library of 98 LMPs to evaluate delivery to the placenta. This technology works through molecular barcoding. So we're encapsulating a barcoded cargo that allows us to track the performance of all of these LMPs individually in vivo, which means we can pool them all together, administer them in a single cohort of pregnant mice, and then look at delivery to our tissues using next-gen sequencing. So this is a heat map schematic depicting this data. Again, this is kind of the great thing about high throughput screening, where you can generate large quantities of data. I'll just kind of point you to a couple things. So we look horizontally, we're looking at all 98 LMPs in this library. We're looking vertically at the different tissues of interest, and of course, darker colors here are greater delivery. I'll just mostly highlight the delivery or the data in the placentas, where we can clearly see that some LMPs are performing better than others, which is really great. This allows us to do some regression analysis to identify a lead formulation, a lead LMP that's going to be placenta-tropic, and perform some validation assays. So I'll show you a little bit of those validation data here, where we're comparing the performance of this placenta-tropic LMP that we identified from high-throughput screening compared to this off-the-shelf FDA-approved formulation. We're using a reporter system, an mRNA reporter system that actually allows us to look at functional mRNA translation using a luminescence readout. So here we're looking at the performance of our liver-tropic LMP. It's performing as expected, lots of liver delivery. When we look at our placenta-tropic LMP, we instead see a lot of spleen delivery, which is actually very interesting and also exciting. It's this idea of maybe achieving extrahepatic delivery. We're escaping the liver, and we quantify this below with the spleen-to-liver ratio. Of course, this is the exciting data here. How did this platform, this drug delivery technology, work in the placenta? We're seeing really promising placental delivery with our formulation. We're outperforming this off-the-shelf formulation by more than 2 orders of magnitude, so that was really encouraging. Now, of course, what do we do with this technology, right? And that's what this audience is hopefully most excited about, is this idea of, can we encapsulate a therapeutic cargo, like a VEGF mRNA, a growth factor mRNA, to treat preeclampsia? Hopefully with this idea of inducing vasodilation. Here are some of our model outcomes that we looked at in a couple mouse models, preclinical models of preeclampsia. Of course, one of our primary model outcomes being, are we actually ameliorating hypertension in these models? So Just high-level overview of one of the first mouse models we did. This is a well-characterized inflammation-induced model of preeclampsia. So this is an early onset model. We're inducing preeclampsia early in pregnancy. On gestational day 7, administering our therapeutic. So this is the placenta-tropic delivery technology, encapsulating VEGF mRNA. We're doing this mid-pregnancy, tracking blood pressure throughout. So these are our primary model outcome data where we can see that just with one injection of our VEGF mRNA LNP, we're completely reducing blood pressure, completely rescuing hypertension through the end of pregnancy. We look at the PUPs collected at the model endpoint, and I hope you can kind of see that the PUPs that are from the preeclamptic pregnancies are very clearly small for gestational age, and that our therapeutic is rescuing this effect, and that's quantified below via litter size and total litter weight calculations. Here we're looking at placental vasculature, right? Is this therapeutic doing what we are hoping it's going to do, which is induce vasodilation in our tissue? So these are H&E stained sections of placental vasculature where the white regions are the actual blood spaces or the blood vessels. I hope you can appreciate the blood vessels are very clearly constricted in our preeclamptic placentas, and we're inducing some vasodilation with our therapeutic, and this is calculated to the right with blood vessel area. Lastly, I just want to show a little bit of data from this hypoxia-induced model of preeclampsia. This is a model that more closely recapitulates preeclampsia as it exists in humans. So we're administering we're inducing overexpression of sFlt1. That's our clinical biomarker of preeclampsia. It's the soluble form of the VEGF receptor. So the model is pretty much the same. The only difference I really want to call your attention to is we're comparing the performance not only of our technology, right, but this off-the-shelf formulation. How do we stack up? And I think this is really the most exciting data from this work to date, which is that we see again our placenta-tropic LNP, the blue curve, we're rescuing hypertension with a single injection. But when we look at the liver-tropic formulation, the pink curve, we're only seeing reduction in blood pressure for about 48 hours, and the mice again become hypertensive, which is really this idea of we needed this rationally designed technology. We needed this LNP to be designed to be placenta-tropic to see the sustained alleviation of maternal hypertension in this model. Just a little bit of data to kind of corroborate this idea. We're looking at serum levels of VEGF. Our liver-tropic LNP is producing a lot of VEGF and secreting it into circulation, but this systemic VEGF is not enough to see the sustained improvements in hypertension. Again, this is quantifying blood vessel area. We're seeing that only with our placenta-tropic LNP are we seeing significant evidence of vasodilation. Lastly, some more images of PUPs to show you the effects on growth restriction. We're looking at serum levels of our clinical biomarker. Again, looking at the gestational day 17 at the model endpoint. Only with the placenta-tropic LNP are we reducing levels of this clinical biomarker of preeclampsia. And then lastly, just kind of showing some liver toxicity data. I mentioned at the very beginning that preeclampsia can cause elevated liver enzymes and liver toxicity, and we're seeing that recapitulated in this model as well. So what have we done? We've utilized high-throughput screening and identified a placenta-tropic LNP. We've shown that this platform can ameliorate hypertension and improve fetal health in multiple models. We've shown that perhaps this tropism to the placenta is really essential for sustained improvements. Where are we going next? Of course, it's a long pathway to the clinic. Here's where we are now, and I just wanted to share some things we're thinking about going forward in my lab. How do we think about placental structure varying across species and how data in different models can actually predict what might happen in a human? How do we think about non-animal models, so organ-on-a-chip technologies or in silico models to really accelerate drug discovery to the clinic? And then, of course, a lot of the barriers we've already talked about today for translation to some of these patients. So I did a lot of this work during my previous training at the University of Pennsylvania, but I'd, of course, like to acknowledge my newly formed lab since we just launched this July. Thank you so much.
Thank you. I'm really happy to be here with you guys. And then I'm going to share what we do. One part of the research that we do in the lab, we work with wearables for health monitoring. Whenever time that we think about wearables, those devices like watch comes into mind. They're pretty good, but they are missing some important molecular level information that we can only have with these point-of-care devices. So our job is to combine those watches, rings, and all the smart wearables with those chemical biosensors. Build everything into one single platform that can monitor your biomarkers in a non-invasive manner, right? I can use anything but blood. We can do contact lenses, for example, for analyzing tears. For being non-invasive is especially important for babies, right? We developed a platform that is a pacifier that collects saliva from babies and toddlers. You are born with type 1 diabetes, for example, and then you can only use the continuous glucose monitor from 2 years old. So even though like other kids younger also they have to do the heel pricking for the blood, right? So no invasiveness is really important for them. Sweat, we work a lot with sweat because it's easy, and then you have sweat glands over the whole body. So whichever like textile or even the socks, we put some sensor on the socks. We sweat a lot. On the hand, your hand sweats all the time, and then we can harvest. Sweat is easy, so this is why I work a lot with them. And then also interstitial fluid for the continuous glucose monitor. They also use interstitial fluid a lot. It's not reading glucose in blood, but in the interstitial fluid through microneedles. So today I'm gonna be focusing more in women's health. There's a huge part of what we do in the lab. And then I'm just gonna share our shot to the moon idea for working with women's health, what we are really passionate about achieving, right? What if we monitor hormone levels, female hormone levels, from the beginning of the menstrual cycle until the end? The beginning and the end is where they go crazier, right? When you are like starting the menstruation, your body doesn't know how to regulate the hormones, and then you see a lot of side effects just because the hormones are going crazy in the teenager. I want to point your attention specifically for this one. Who here has like a teenage daughter and haven't heard yourself talk so many times? My kid is not interested in anything. She doesn't do anything. She just sit on the couch and do nothing, right? She's really lethargic, and then it does like probably. She might be having like heavy menstrual bleeding. This is not a conversation that she has with friends or with parents, right? But it can develop like really severe anemia. And then it is preventable. If you know like how much blood you're losing during the whole month, you can catch it before that appears in a blood work, for example. If you see in a blood work that your anemia is there, you're already like too far into it, and then you have to take iron into the veins and such. So monitoring hormones is important since the beginning of where they are appearing in the body, right? And then throughout the women life for reproduction, and then even like a regular, just by being a woman, there's other hormones and symptoms that are there, and they're really important. But what I think it is really amazing that this polycystic ovary syndrome is so common between women, but people don't know why they are there, or like how they go away, or how they get worse or better. So little is known about everything that happens in the women's body related to hormones, right? Our goal is just to understand what is happening to your body when your hormones are fluctuating so much. Not only during, for example, if you're looking for a fertile window, but during pregnancy and after pregnancy, there are like the fluctuations of hormones that matters a lot. To be, what is that? It's gonna turn off? What did I do? What happened here? All right. Oh, it's only my screen. Yeah, you guys don't see. All right. I'm going to lose my slides. All right. So for this particular slide that you can see here on the table, our lab is really interested in preeclampsia as well. So we do a lot of like wearable blood pressure monitoring. When you have preeclampsia, women have to stay in the hospital and monitor the blood pressure like very often. Or even after birth, they can develop preeclampsia, and then the severity is even worse because now they are busy with baby, they are busy with life, and they're not gonna go back to the hospital to be monitor their own health, right? if you're feeling bad, tired, or anything, you're just power through. This is what we do. You feel down, and then you just go with it, and then take care of whatever you have to. So monitoring the hormone is going to give you this clear idea of what is happening and why it's happening, so you can fix. It's fixable, right? This is important. And then our goal, our dream, is to make an implantable device that can monitor metabolites, hormones, and other ions, and whatever is important, not only for women, but for health in general, that you can wear from the beginning until the end of your reproductive life. And then we call it BEAM, biomarker and endocrine analysis for pregnancy and health. We have started doing some preliminary work towards this device that would be implantable and then stay in the body for a long time. The longest that I'm aware, those birth control, that it is under the skin, it stays there for 5 years. So this is my goal right now, just because people have done, why not? So just put the sensors over there. What we are doing now is a small pilot study that we have some women's that we are gonna be monitoring their menstrual cycle for 2 months, 2 cycles. Every week, we collect sweat, blood, and saliva, and we do some cognition tests. The hypothesis that we are trying to answer is there a correlation between cognition, how fast you can think, and then correlate, numbers or ideas, how fast? Does it depend on your hormone levels? In this case, we are hypothesizing that the progesterone is the bad guy, so whenever it goes up, your capacity of thinking fast goes down. So at the end of this study, we are gonna have a lot of molecular data from all these biomarkers plus wearables, watch, the ring, everything that we can think of, and then we have a huge molecular data, as we saw in the beginning, and the wearables that is already out there, and then we can study if there is correlation using machine learning. The goal is, at the end, maybe you won't have to do the molecular tests so often. The wearables by itself, they're gonna be able to make these predictions, and then you can just calibrate your system with, once a month, twice a month, actual biological testing. So this kind of study has been done before, and then the conclusions were yes, there's correlation, no, or inconclusive. So what is different this time? What I'm doing this again is just that before, we didn't have the technology to monitor the hormones that often and with such precision. It was just based on self-report. When are you in your period? You know how long it lasted and your symptoms. It was not, the molecular data was not there. So what we do, we give this kit for our volunteers to monitor the hormones in urine, blood sugar with the glucometer temperature, and then at the end, they log all the data. These are, I think only the Pregmate is the FDA approved, so we are comparing other tests that is already available out there with that one. Those are done in urine. We also have data on blood that we are collecting in sweat and saliva to try and make this easier, because this takes a long time to do. Imagine you wake up in the morning collecting urine and waiting for 10 minutes for one test, 10 minutes for the other test. The goal here is to combine, remember in the beginning, all of these hormones and molecules and analytes in one single platform that you can just stick to your body and forget about it. So you don't have to have so many lancets and then alcohol and then another device. Just put that and forget. This is initial work that we did just by looking into estradiol in sweat. These are 2 female subjects looking into sweat in the bottom compared with blood in the middle, and then urine at the top. So you can clearly see the 2 menstrual cycles rising and down of estrogen. The goal now is to make it multiple sensors with estradiol, progesterone, cortisol is really important, and we saw highlighted on the table, and glucose itself. So yeah, that is what we are trying to do, and then our shot to the moon is maybe have a better understanding of our own health, and then we can have better control of it. Thank you.
I'm going to follow up my students' talk earlier during the lightning session or fire talks with a little bit more description of our technology that is used to deliver vaccines and thinking about how we can use that to really impact a large number of people So vaccines are incredibly useful and also incredibly cost effective, which is important for access. Obviously, we've saved over 150 million deaths since or 150 million lives since the beginning of the World Health Organization's expanded program on immunization, which started in 1974. And so this is a great success of human healthcare in history. Also very cost effective. For every $1 we spend in a low and middle income country, we save $52 downstream. So really much easier to prevent than to treat, of course. However, we should not pat ourselves on the back too much because infectious diseases are still the second leading cause of death worldwide, behind cardiovascular disease and actually ahead of cancer. So we still have a long ways to go. Additionally, because infectious disease tend to predominantly affect younger populations compared to maybe a cancer or a cardiovascular disease, it's a leading cause of disability-adjusted life years lost worldwide. And so there's a couple of reasons that we aren't doing a better job in this space. One is a lack of access, and one is a lack of good vaccines, right? And so every year there's almost 20 million infants that don't have access to standard vaccines, right? And this number was going down for a long time, and now it's unfortunately going the opposite direction. So it's not going to just address itself. We have to come up with interventions, whether those be social, educational, or technological. Obviously, our lab is focusing on the latter. And then other things, we just don't have good vaccines. So malaria, for example, there is a vaccine for malaria. It just doesn't work very well, right? So you have to give it 4 doses at different times, and you have to, and then you only have less than half the population is protected for a limited amount of time. So we really need these robust vaccines that are, give them as an infant and then maybe come back a decade later, or ideally never come back, right? And so we're trying to develop vaccination technologies that address both of these needs. Can we make vaccines that have maybe failed or are infeasible to implement in the clinic now feasible to implement, right? And also, can we reduce the number of times? Oh no. Okay, don't worry. I got it. Yeah. Just keep you on your toes. Okay. So, or think about how we can minimize the number of times we need to actually see a patient in order to prevent infectious disease in that individual. And so ultimately, 1 dose is typically not enough to induce long-lived immunity. The reason is because our immune system has to allocate resources to build that immune response, and it doesn't make a lot of sense to every time you see one pathogen one time to commit a lot of resources to protecting against that in the future, right? But as you see it more and more, then it becomes kind of a note to your immune system, hey, pay attention to this. This is going to be in the environment for a long time. And so we have to give multiple doses, and so you might see something like this if we think about humoral immunity. The antibodies that we want to generate when we give a vaccine are these neutralizing antibodies. These are the ones that prevent the pathogen from infecting you, right? And so maybe after 1 dose, you're not over that kind of immune threshold. Then another dose you are, but you would kind of be below it after a little bit of time. And so we really want to boost this as high as possible. And so our strategy is to, instead of giving multiple injections, we can give 1 injection of a biodegradable microparticle containing our vaccines. And so we want to engineer these particles to give us the same kinetics that we know to be safe and effective with N equals billions over the last 50 years, right? And so we want to have these pulses come out at the same time as a vaccine would normally be administered. Again, that's a 1-month spacing in low and middle income countries where you really need to vaccinate anytime you can see them and it's appropriate, or maybe 2 months in high income countries. And so the problem is that we're not some geniuses that have developed a new idea here, kind of. This has been an idea for a while. How can we make these single injection vaccines? The problem is the tools haven't been available. So a lot of people make particles that look like this. There's a dozen plus FDA approved delivery systems that deliver drugs, not vaccines, and they have this kind of structure. And so the problem with this kind of structure is that you get a release curve that looks something like this. So you get a lot of your vaccine released early on, and then progressively slower and slower amounts of release, right? And so this is actually exactly what you don't want for a vaccine. You are telling your immune system that whatever you're doing, the pathogen burden is going down, right? Because there's becoming less and less of the antigen present. And so we hypothesized that we could have a different structure, this core shell structure shown here, and then we could have kind of an all or nothing release event. So in the left case, we see there's a bunch of different pockets. All those pockets get exposed at different times. We want a single pocket so that we can have this all or nothing effect. Early on, even if we use the same biodegradable polymer, we get no release because we haven't created enough porosity in this material to form a channel until we get to the point where there's, oh man, I'm not good at this. There's a critical release path, and then everything comes flushing out, right? And so that's the hypothesis of how we could achieve this kind of kinetics that would give us a burst. And then we can combine multiple populations in order to get a second burst and a third burst so we can give all the doses needed for a particular vaccine or maybe even multiple vaccines. Unfortunately, there wasn't the technology to make particles with this kind of core shell structure, so we developed the pulse method, which my student Catherine alluded to before, and she kind of explained that we mold these little buckets, we fill them with a little robotic dispensing machine, and then we can seal them using a little bit of heat, and they wind up looking something like this. Here's our polymeric biodegradable buckets filled with our material of interest, usually going to be vaccines. In this case, it's a dye to see it easily. And then we kind of have this snow globe structure at the end, which we can then inject. This is how they look at different stages. So here's our filling process. So we have this little dispenser that can go down there and fill our little tiny buckets. If we look from the top, we see that we can precisely hit this large arrays of, in this case, cubic particles. But nevertheless, we can fill them very precisely with the amount of vaccine or excipient that we need. Then we want to seal them. This is a real-time video of a sealing. I want to direct your attention to these yellow arrows. Hopefully you can appreciate that it's initially in a right angle, and then over time, the material at the top of this particle starts to flow as we raise it above its glass transition temperature, which causes it to seal the top. So we're actually using the material that's initially at the top of the bucket to cave in on itself and create these snow globe structures. These can be miniaturized, so this is a picture that we really like in the lab, obviously. We can see that we can miniaturize them very small. Here's the largest type of particles in an 18-gauge needle. We made smaller particles that can fit through potentially as small as a 30-gauge needle. We'd love to be at 25-gauge or smaller in order to be on par with current clinical standards for infant vaccination. And so, of course, the question is, do they work like we want them to work? Unfortunately, the answer is yes, they do give us this delayed pulsatile release, and we can tune that release from really days to all the way out to months. And the way we control that is actually not with the thickness of the wall, which is a question I get a lot. It's with the properties of the polymer. So in this case, we're only using one material, or the data I'm showing here is only one material, polylactic co-glycolic acid, PLJ. That is what your resorbable sutures are made out of, right? So this has a long history of biocompatibility. I think that was a question from the audience before. So people generally feel very safe, or very good about this material, and we can change the molecular weight, the copolymer ratio, and the N group in order to kind of titrate when this delay happens. Here's what these particles look like at different times in the process. And so early on, you see we have these little snow globes, and then they start to swell, and then they start to release, and we can hit all sorts of times, right? So here's kind of examples of different pulsatile release that we can achieve from whatever, a week to a couple of months, right? So hitting these vaccine-relevant time points is really important for us. And so again, in vivo, they also work. A lot of things in the controlled delivery space fail at the in vitro to in vivo conversion. So fortunately, ours does not. In this case, we're filling these particles with a dye and releasing it and seeing an increase in the amount of fluorescence. Initially, the fluorescence is quenched inside this tiny particle, and then it spreads and increases a huge amount in order to be able to detect when that comes. We again see that we can get this delayed pulsatile release, and then we can tune it based on the material composition that the shell is made out of. So we wanted to see if this works in general in a vaccination model. So we used a model protein, ovalbumin. This is literally the egg white in your chicken eggs. And so we delivered multiple particles that we're going to release at different times, and I'll just briefly walk you through them. So if we give one injection of two particle populations, one that released in 9 days, one that released in 41 days, we see this kind of response. So we were trying to get non-inferiority to 2 injections delivered at the same time. When we do give 2 injections delivered at that 9 and 41 days respectively, then we actually see a reduced response, right? And so this was curious to us. We actually weren't thinking that we were going to beat it. We were thinking we would match it and then save on the number of times we need to inject. So we wanted to explore this a little bit further. Seems, I'm not doing well with this. All right, there we go. It seems like one of the factors here is that our polymeric particles do act as something of an adjuvant, and ultimately the effect of that adjuvancy plus probably some benefit of our control release, which is pulsatile, but not as pulsatile as injecting a soluble solution, is that we get about a 2-fold dose-sparing response. But the next question is it harmful to the protein? We need to deliver vaccines, which are generally protein or some type of an activated virus also made of protein, in its stable conformation, right? If we have it change conformation, then we are building immunity against something that doesn't infect people, and we need to have those neutralizing antibodies, right? So these are alarmingly hot temperatures that these particles get to, and so we wanted to see, are these really bad for the patient? I don't know what I'm doing wrong. It's not going well for me. We wanted to see if we were going to have this loss of stability. And so in some cases, depending on the protein we put in there, we see that there is major problems. In other cases, it's actually fine to fill and even seal, and they're okay. But the problem really happens at the point where we're releasing them, right? So these are going to be in the body for a month or 2 months, and they are going to be subjected to 37C. We can't change the temperature of your body, and we also possibly have other insults. So these PLGA particles degrade into lactic acid and glycolic acid. Lactic acid is what your muscle is making right now when you move it, so very widely considered biocompatible, but nevertheless, we see really major stability problems in this case. If we put in this antibody, for example, we see 0% is still stable inside that particle after just 2 days, and we need to achieve months, right? And so we saw with excipient formulation, we added things that stabilize inside the particle. We can actually get very good release. This is a cool assay where we cut the particles open and look how much is still inside them or how much is released, and we can see the amount inside drops down as it gets released. The amount outside comes up again as it gets released. And so we now are applying this to different infectious diseases, namely rabies. Rabies tied for deadliest disease in the world with a 100% fatality rate. You really need to intervene before symptoms occur, right? And so there's kind of a two-part treatment here. One is the monoclonal antibodies. Those protect you immediately when you deliver them. And then you also deliver vaccine doses at 0, 3, 7, and 14 days. And this is, a major accessibility issue, particularly in low-resource settings, because it's hard to go to a clinic not only one time, but 4 times now within a couple of weeks, and you don't know if you actually have been exposed, right? Because that animal hasn't been diagnosed to have rabies. So in short, we've stabilized the kind of contents in here. Dispensing didn't do any damage. We figured out how to seal them without doing damage, which helped us lower the level. And now, eventually, through multiple iterations of these excipient formulations, we can find formulations that are able to keep enough stable to give a dose that would be protective. So in our rats, we need one of one type of particle, one of another type of particle, and 2 of another type of particle in order to deliver a sufficient dose to be neutralizing. So we think about lots of different ways to deliver this, either more doses to make vaccines more effective without adding feasibility challenges, kind of co-delivery of different vaccines and offset patterns to get the full response to both of them, and then playing around with kinetics in order to get even better responses. Again, because we know vaccines are hard to develop, we are trying to use our abilities in the control-release space in order to make these work better, right? So hopefully convince you that we have some technology here that can hopefully act as a single injection vaccine someday sooner than later, and now we're just trying to improve upon this, particularly in the stability space, and then building out our library fully. So with that, I'd like to thank my team and all of you for listening, and appreciate any questions during the panel discussion session. Thank you.
Good afternoon, everyone. It is a pleasure to be here. I'm really happy to share some of the innovations we're developing in my lab at UNC. And this is such a fitting theme and conference for it, and glad to be here in the inaugural meeting, hopefully one of many. These are my disclosures. So just wanted to start by sort of highlighting the global women's health market, and the good news is that it's continuously increasing. The bad news is that women's health is highly underserved, underinvested, underfunded in many, many ways, and things hopefully need to be changed. In my lab, we focus a lot on women's health. We have different technologies. We are targeting different unmet needs. A lot of it is focusing on infectious diseases, HIV, STIs like herpes simplex virus, and unplanned pregnancy, trying to develop first-in-line multi-purpose prevention technologies that can hit 2 or 3 of those unmet needs. I have started a company, Anello, out of my lab using a technology that's highly innovative that I'm going to talk about in a few minutes. And we're targeting our first product for infertility treatment, developing a long-acting release of progesterone to supplement progesterone for women who undergo infertility. We have also a thin film technology for vulvodynia and many other technologies in the pipeline. So for those of you who are not familiar with statistics for HIV and unplanned pregnancy, these statistics speak for themselves to highlight the huge unmet need globally for both HIV infections and unplanned pregnancy, both in the US and globally. These are infections that are continuously increasing, and unfortunately, due to the current climate, I anticipate that these statistics will continue to increase dramatically. And so there is a significant need, if you look at the landscape for HIV prevention, pre-exposure prophylaxis, or PrEP for short, and contraception, there are multiple different options for contraception, long-acting, short-acting. For HIV prevention, for many, many years, the only option was daily PrEP, so a daily pill that people that are at high risk have to take daily. So you can imagine once they miss a pill or 2, the infection risk increases dramatically. However, in the recent years, this year, once every 6 months injection was actually approved, lenacapavir long-acting by Gilead. This was a high breakthrough. Another long-acting that was approved 2 years ahead of it was Apretude, and that's a cabotegravir by ViiV that is given every other month. So there's a lot of breakthroughs in long-acting, and the field is continuously pushing for longer-acting because of the high unmet need for these technologies. However, to date, there is no long-acting or any multi-purpose prevention technology that can actually prevent HIV and unplanned pregnancy in one product. The only product that's available is the condom, and it has many, many different limitations, as you all know. There you go.
You're the lucky one.
That's Kevin. I called it. Oh, there we go. Awesome. Fantastic. So my lab has been for many years developing long-acting delivery systems. We have a first-in-line ultra-long-acting. By ultra-long-acting, I mean 6 months or longer, injectable that's being developed for HIV prevention and unplanned pregnancy. So that's one shot to deliver an anti-HIV and a hormone. We have a novel biodegradable implant that actually we developed a new manufacturing process based on the long-acting injectable, knowing its limitation. And then we have this innovative 3D printed intravaginal ring technology, or IVR for short, that we've been developing as first-in-line MPT, or multi-purpose prevention technology, to expand options for women and young girls in low and middle-income countries, or LMICs. And so we're using the state-of-the-art 3D printing, and you're probably going to think this is science fiction. It's not, I promise. And it's really the technology that I started a company out of it, and the company is developing the product for ART. So you may ask, why are you using 3D printing? There are intravaginal rings that are in the market. There's NuvaRing, FemRing, S-Ring. They're all made by the same exact process, and this is where it goes by. When I say women's health is underserved, underfunded, and underinvested, it's really stuck in the 80s as well in terms of product and engineering technologies that are used to develop new technologies for women's health. So the only technologies that are out there for women's health that produce intravaginal rings are injection molded rings. These are fairly limited in terms of the materials that are used, the properties of the ring, the drugs that can be formulated in them, just because these processes inherently use high heat, high pressure, so you're limited in terms of the drugs that can actually withstand those high heat and pressures. So we have actually leveraged, it's not you. I believe you. Is it connected? Yeah. Okay. So we are using a state-of-the-art 3D printing. 3D printing has existed for many years now, but this is a continuous process. It's called continuous liquid interface production, or CLIP for short, or digital light synthesis, or DLS. And what it does is actually really clever. As a polymer chemist, for those of you who do free radical polymerization, the first thing that you want to make sure is to not have any oxygen in that vessel, otherwise it inhibits polymerization. And so what the company Carbon, that owns this technology, did is actually leverage that inhibition by oxygen to introduce what they call this dead zone, in which the polymerization doesn't happen, and above it, the polymerization happens and solidifies the part, and it makes that process continuous, which makes the 3D printing highly throughput, highly scalable, commercially scalable, and produces parts that are layerless, monolithic, soft, and if we're talking an intravaginal ring, the first thing we want is a soft material and a soft product. And so this is a 3D printer in the lab scale. In the company, we have twice the throughput. This printer allows us to print 16 rings at once. In the company, we have 32 rings at once. And then we have it, this is a scalable product process where we now have GMP manufacturing that can actually connect 2 or 3 printers or more to have a chain production that's commercially scalable. And so why 3D printing? We have now the freedom to introduce intricate features because we use computationally aided design, or CAD, to introduce these designs that cannot be introduced by traditional injection molding or extrusion. So instead of using those solid cross-section that looks like a cylinder, we're now having these features that allow us to completely fine-tune the release kinetics and the duration of these APIs. Not only that, but we actually incorporated a new way by adding the drugs into the device post-fabrication. So we're not exposing the drugs to the 3D printing, the high heat or pressure. And for the first time, we demonstrated that we can release the entire payload that we load in these rings. Unlike the injection molded rings, where you actually release at best 20%. And so we can save a lot of money. We have rapid development from design to final product based on a target product profile that takes months instead of years. And so we've done a lot of work to, this is just showing you, I'm not gonna belabor this process, but we start from a solid torus that looks just like an injection molded ring. We implement what we call these unit cell designs with CAD, and then we 3D print the design that we are targeting for a specific drug. And because we have a high throughput with CAD and 3D printing, we were able to do a lot of focus groups in North Carolina to actually talk to the women who are our end users to look at what they prefer in terms of the look, the design, the size, the color. And we took this all the way to a second in-person as well as over the phone. And then most recently, we had funding from the Gates Foundation to take this, these focus groups to Zambia through UNC Zambia collaboration. And this study just ended, and we're not analyzing this, the outcome of it. I'm very excited to publish it soon. So with this technology, we've been making these first in-line 3D printed MPT. We have one that we recently published on to combine an anti-HIV and anti-herpes and a hormone. And we took this all the way to non-human primates and showed feasibility and PK and safety of this very exciting technology. And then another one that I'm really, really proud of is this first in-line MPT for HIV and contraception that combines a highly potent anti-HIV drug, islatravir, with the NuvaRing hormone combination, EE and ENG, to make this new MPT to deliver these three drugs. And we load all three drugs in a single step process. We have shown, and the one thing that I haven't really mentioned is that the anti-HIV drug is hydrophilic, the hormones are hydrophobic, and we are able to actually combine these three APIs with completely opposite physical chemical properties in a single step process and release them in a controlled way in a steady state release. Not only that, but we were able to actually show that we're hitting the right PK in non-human primates, the right PK compared to the NuvaRing and sheep. In the sheep model is actually the perfect model because we are for PK and safety because we use the human-sized rings. In the macaque model is a great model for HIV because we can infect them with HIV and look at efficacy or prevention. And so we've done a lot of work with vaginal microbiome to look at the health of the vaginal microbiome, making sure that these MPTs do not harm the vaginal microbiome, and demonstrated that they're really well tolerated. And so we took this MPT, which is designed to be the first 90-day MPT that women would administer once. It lasts 3 months, so 4 rings to protect them against unplanned pregnancy and HIV with 4 rings per year. So in terms of sustainability, that's 4 years, that's 4 rings instead of 12 rings if we were to develop a once a month ring. And so we've demonstrated through a crossover PK safety and a 90-day PK that we can release the islatravir, the anti-HIV drug, over 90 days at target therapeutic levels. And then we did an efficacy where we took the macaques and challenged them vaginally and for these with the islatravir rings or the anti-HIV ring, and demonstrated full protection for the first time with a single administration of a ring in non-human primates that protected against 12 weekly exposures. And we followed actually the macaques post-ring remover to make sure that the HIV was not just suppressed, and demonstrated that they were fully protected. And so this was very exciting. As I said, this is a platform technology. My company is developing it for releasing progesterone over 4 weeks so that women can actually administer progesterone independently to replace the painful shots that they have to take or be administered by a partner for assisted reproductive technology with only 2 to 3 rings. And we're expanding this to other indications such as menopause, gynecology, oncology, and many other indications. With that, I'd like to thank everybody that does this work, collaborators, the funding. Thank you all for your attention.
Kelsey, I wonder, could you talk a little bit about whether or not it would be useful to be able to directly image blood flow in the placenta? Would that be helpful?
Yes, definitely. I think there are definitely ultrasound imaging techniques definitely to look at blood flow. I think that's where the preeclampsia field struggles, is this idea of early detection and how maybe a therapeutic like what we're trying to develop could be used to intervene before long-term onset. But definitely there are people working on these kind of blood flow imaging techniques in the placenta. Obviously not my expertise, but it would be really interesting to integrate that with some novel therapeutics, definitely.
I have a question for Dr. Sempianato. Hope I said your name correctly. What do you expect that women would do with this information about their hormones, and how do you think that your results or your hypothesis, if you're correct, that women's cognition changes with their hormone levels, do you think that that could result in like stigma against women in workplaces and things like that?
Yeah, that's a really good question. There's like a pros and cons. Actually, why we started to do this study was because of a conversation we were having. Women were talking, oh, sometimes I develop some allergies, and then I don't know when or why. Or sometimes I have really, really bad brain fog, and then it would be nice to have like some heads up that this thing is about to start or to understand. And then there's always the conversation, oh, I'm taking this, that it worked for me, oh yeah, I'm gonna try to. And then there's no like a test of anything. And it's just like trial and error, like this supplement or this vitamin works for me, but there's no actually research on what medication we should take, and then what is the period of because when you are into menopause, the cycle and the fluctuations of hormones are unpredictable, and then you just don't know what is gonna happen with you until it happens, right? So having the ability to do prevention care, it would be the best to do with the information. And then the risks, that I was thinking a lot about that too. Let's say we did an awesome job, and then we could correlate cognition with the hormone levels. Now you are the employer contracting for a high risk position, like a airplane controller traffic, And then would you hire women over 40 knowing that their cognition is gonna decrease because of their hormones? Would you do that? So this is one of the risks that this research might imply, and also people might over-rely on this information, not going to the doctor, self-medicating, and then, I know what is going on with my body. I have the control now. So those are all the risks. But I believe the benefit is not only for what you are going to do with the information, but what we're going to learn with it. We don't know why hot flashes happen. We don't know what polycystic ovary happen, Is more benefit so far than the risk. Stigma, I am also really concerned. I keep thinking some women from the future is gonna come back to destroy my research because women is not hired for anything anymore, So yeah, I keep thinking about that too. If that happens with you and then you cannot prevent.
For Dr. McHugh about your vaccine and your team vaccine innovations. So my question is about our current social climate, about how more people are more distrustful towards vaccine now, and more people want to take less and less vaccine. And I just want to ask how you and your team would combat against that, because you are making a new technology about enveloping the vaccine and make it at a later date, so people are more distrustful about putting that in their bodies. So I just want to hear your thoughts on that.
Yeah, sure. Definitely a big problem. Maybe in some regions of the globe more than others, particularly those who have used vaccines to great success at locally eradicating some diseases, right? So we have a lower actual and perceived risks in some high-income countries, right? So other places, I think, are much more open to vaccines because they see infectious diseases causing problems on a day-to-day basis. I don't think that a technological solution like the one we're trying to develop is necessarily the only thing that we have to do, right? So I think, education of populations is super important. Maybe I'm still an optimist in that eventually we will come back to the area where we, make science-informed decisions about health. But I think it's not something that technology alone can deal with. So hopefully we have some folks, maybe like those that are in the audience here, that have, say and can do some of that kind of communication of the real benefits and perceived risks of vaccines, including with our technology.
Thank you.
I came in late. I had to take a call. I apologize.
You're pointing at me.
Your ring. So, if this has been talked about, forgive me, but is there a possibility this could be also used for postpartum depression in the event that one even has that kind of run out in their family? This could be on tap after child delivery, because 48 to 72 hours after, those high doses of progesterone and estrogen, they fall, and then your serotonin and your dopamine, they lack. So you go into this, and that seems that this would be ideal to prevent that.
Thank you for that. No, we have not thought about postpartum depression, but that's something that we definitely should actually explore. It's definitely the hormones are things that we're delivering with the rings anyway. So it's just a matter of understanding. It's not just local delivery, it's systemic delivery as well, just like NuvaRing. But we are, I think the main, having a company made me realize what it takes to actually start something and take it all the way to commercial. And so we're trying to get, trying to get my lab staying focused on the products that we have advanced all the way to non-human primates and get them to first in human, and understanding the regulatory, understanding the market, understanding the policies, understanding how to implement them. It's a whole other science that if I didn't have the company, I would have been very naive to what it takes. And so we're trying to stay focused, but we're trying to also look at the opportunities and the unmet need, which is why not cautiously, but strategically expanding the platform without diluting or staying away from advancing the products that are already further or closer to first in human to start making impact. But that's something that we're definitely going to explore. Thank you for bringing it.
You're welcome.